So just to finish up here on the Casparian strip, we said it was a layer of suberin that impregnates the cell wall of the endodermis, which you can see in the enlarged figure here, the purple that basically encircles each of these cells, leaving those longitudinal walls free there so that water is guided from between the endodermis cells by way of that apoplastic transport or route across the plasma membrane. So that basically creates a resistance for water flow or transport. And so one of the resistances that we could note is the presence of the Casparian strip for guiding water through the endodermis and into the xylem. Interestingly, the Casparian strip is also going to prevent water from moving in the opposite direction and also help maintain that higher solute concentration inside xylem, which maintains its lower water potential. Again, allowing water to move towards the xylem down the water potential gradient. So the Casparian strip is important in a couple of ways there. Including maintaining keeping what's in the xylem there. Another resistance that we've already mentioned as well, but just to make note of, is the physiological resistance. Let's see, let's get that right. Which is the semi-permeable membrane. So that, as we said earlier, prevents everything from coming into the xylem, but it also kind of slows down the movement of water. So there is a physiological control of what enters the xylem. One last thing to mention in this lower right corner here, which is where fluorescent dye or radiation is allowing us to see the micrograph of a radial section of the root or a cross section of the root. We can see the green highlights the presence of suberin here in the endodermis. We can also see those thickened secondary walls of the xylem in the vascular cylinder there. But we also see suberin in those older regions here of the epidermis. So suberin is, showing up in the epidermis as well, where we said that the movement of water is more difficult through the epidermis when in those older regions of the root. So now that we see root has entered the xylem, then we can now refer to how water is transported from the root xylem up through the stem to leaves. Okay. So to talk about that process, we want to basically review what is the structure of xylem. Okay. Xylem is continuous between the root, the stem, the branches, all the way up to the leaves. And so the components of xylem include the sclerenchyma fibers that we saw when we were looking at plant anatomy, that are fibers that are just to the exterior of the vascular cylinder. And they contain those secondary walls that help support the structure of the vascular cylinder and therefore provide, some structural support for the whole plant, kind of like the human skeleton. Another component of xylem includes parenchyma cells, which are for storage and transport. And remember, they only contain primary cell walls, so they're not so helpful for support, structural support, but for supporting xylem in general. And then the component that we're going to spend the most time talking about are the tracheids elements. Let's see. The tracheids elements, which are vessel elements or members plus tracheids. And that's what we're looking at diagrams over here on the right. Now, tracheids elements have several things in common. They have the lignified cell walls. They typically have some secondary cell walls too. The lignified cell walls makes these vessel elements and tracheids impermeable, the walls impermeable to water, because water is conducting up through them, not outside of that cylinder, that vascular cylinder. It also makes those tracheids elements rigid. Lignin is not only impermeable to water, but it also creates some rigidity, and that rigidity helps to prevent collapse of the xylem or the tracheids elements under negative pressures or tensions, a high degree of tension rather, singular. We're combining words here. Okay. Which is what happens, which is the state that the xylem is in under when transpiration is occurring, and that's the abbreviation here that we're going to use for transpiration. And again, the rigidity also provides structural support as well, so that plants can stand upright like a skeleton, supporting those softer tissues. Alright, so we're going to take a look at the two types now of tracheids elements, and starting with tracheids. Tracheids are evolutionarily older. They are found in, of course, vascular plants. Xylem is only found in vascular plants. So starting with ferns, if we're looking sort of evolutionarily, mosses would not contain vascular tissues, but starting with ferns, we have vascular tissues on up through gymnosperms and angiosperms. So tracheids tend to have these tapered shaped cells, and they contain a diameter of 10, somewhere between 10 and 50. This is just kind of a general range, micrometers in diameter. And they transport water through their pits, or where the pits are aligned as pit pairs, side by side. And so we can see in the black line here, which I'll outline, basically water is zigzagging up through adjacent tracheids through pit pairs, like we see here. Alright, so then we compare that to vessel elements, and vessel elements appear later. They're more evolutionarily advanced, and so we find those in angiosperms. And I believe there actually are some ferns that have vessel elements. There's always some of these exceptions. So we're looking at diameters on average. There's a large variety of diameters that we can see in the literature, but 40 to 60 micrometers, perhaps up to 300 to 500 micrometers in oaks. And they can vary in length, much longer than tracheids as well. But we're not going to, we're not memorizing these numbers. We're using the numbers to compare for relative differences. Water is transferred from vessel element to vessel element through perforation plates. And those are found here in the end walls here of the vessel elements. So these little slits, or a net-like pattern of openings that allow water to flow straight through. So these would be stacked end to end, looking more like a long vessel, or like a straw of a variety of cells here. Alright, so we can use these diameters to reflect on or to ask the question, which would have a higher bulk, or let's see, let me which would have a higher rate of hydraulic conductivity. So would that be vessel elements, or would it be tracheids? All right, so think about that as you look at the diameters there and you reflect on which of these is going to have a greater degree of bulk flow versus something that we'll be talking about shortly, which is capillary action. Okay, so make sure that you take a little moment here to reflect on those, how the diameter translates into the rate of water movement through these, and how water is transported primarily through these different tracheary elements. So we'll take the topics that we just where we left off here, then with movement of water through xylem in the stem and up to the leaves from the roots, and talk about how the pressure gradient that occurs between roots and leaves is going to be overcome to provide the means for water to move up to leaves. So that'll be what we discuss in our next recorded lecture.